Good morning to doctors and all my friends. Today I'll be sharing a forensic case that uses mitochondrial DNA marker to solve crime scene investigation. On the February 1998, a plane from the Philippines, Cebu Pacific Flight 387 was crashed into a remote mountainside of Mount Sumagaya. All 104 passengers and crews on board were killed. This is a Philippines domestic flight departed from the Manila International Airport and was scheduled to arrive in Lumbia Airport of Cagayan de Oro City. So in this case, the identification of victim will be difficult due to the severe environment itself, preventing us using conventional identification methods. The severe magnetic impact and heat stress suffer from the victim, resulting in fragmentation of bodies and severe or mild degradation. The biological evidence collected from the crash site typically very low concentration of degraded nuclear DNA. Hence, the disaster victim identification team has subjected samples from the crash site to undergo a combination of mitochondrial DNA and short tandem repeat STR DNA profiling analysis for victim identification. In this video, I will more focus on mitochondrial DNA marker identification. What is mitochondrial DNA? Mitochondrial DNA is a small chromosomal DNA found in mitochondria of the cell. One of the very important point is that mitochondrial DNA is maternal inherited. It allows scientists to compare mitochondrial DNA profile of the remains to the reference sample to any maternal linkage individual, such as mother, brother, or sister. The mitochondrial DNA profile should be same because all the maternal relatives inherited the same mitochondrial DNA. What is mitochondrial DNA analysis? It is a process that determines mitochondrial DNA sequence from a very trace amount of degraded sample and compare with reference sample from the possible relative to determine whether there is a match or unmatched. The advantages of using mitochondrial DNA compared to STR analysis. Mitochondrial DNA have high copy number, typically thousands of copies in one cell, and its stability. It is sensitive in degraded samples, and mitochondrial DNA profiling procedure with close relatives are not available to use as a reference sample. Any maternal relative is possible. So in mitochondrial DNA analysis, the first step we do is to DNA extraction. Generally, DNA extraction can be done from muscle tissue, blood samples, and bone tissue using phenylchloroform extraction technique. In this case, muscle and blood samples will be using Rujukan genome DNA extraction kit. For bones, a handheld drill is used to obtain the bone material, then transferred to the tube contains DNA buffer. We incubate them for 18 to 24 hours at 50 degrees Celsius. The resulting solution will continue with phenylchloroform extraction process. Both samples extracted quantified using quanti-blot kit to determine its purity. Next, we go into Pcr amplification. Pcr amplification allows production of many copies of regions mitochondrial DNA interested. Pcr amplification performed using DNTP specific primer tag DNA polymerase, 3HCl, MgCl2, and others. During PCR process, the specific hypervariable region of mitochondrial genome are amplified. So next, we continue with mitochondrial DNA analysis. In this case, the distal portion of hypervariable region 1 was amplified using primer pair LB19556 and H16431 primer. And also, the distal 200 base pair portion of hypervariable region 1 was also amplified using primer pair LB16208 and H16431 primer. The PCR process was carried out using a hybrid homogeneous PCR machine. The reaction initiated at 95 degrees Celsius consistent for 5 minutes to complete 30 cycles. This is the denaturation step. The reaction continued with 45 seconds at 50 degrees Celsius and 1 minute at 72 degrees Celsius for the annealing step. The PCR process ended with 20 minutes at 72 degrees Celsius for extension step. The PCR product was made single-stranded for sequencing. In sequencing, all sequencing reaction using Sequenase 2 sequencing kit. We analyze on a polyacrylamide gel and followed by a Fuji X-ray frame to see the mitochondrial DNA sequence. After that, we continue with matching analysis. In this step, a blood samples from the relative was obtained in order to identify the victims. We focus on the maternal relative. This allows us to have an initiator matching using mitochondrial DNA profile. We allow mutation in the mitochondrial DNA when observed at a very high relative frequency. One mismatch is allowed permissible to reduce the possibility of false exclusion. Once there is a match, we continue with STR profile of the victim and relative are compared. So now we have come to our final step, is the identification of victim. The aim of mitochondrial DNA and STR analysis was to group the samples that came from the same individual. So as you can see from the diagram, there are 104 victims from the pain. 187 tissues and bone samples were collected from the crash site. By using mitochondrial DNA profile sequencing analysis, 55 different groups was possible as sorted out. We continue with STR analysis, subdivided sorted group into a total of 80. However, some of the samples in the 80 groups were not compatible. Hence, this finally leads to a 95 distinct groups sorted overall after we compare with post-mortem data. Some of the limitations in mitochondrial DNA analysis are the initial 400 base pair spanning region in the hypervariable region 1 was initially amplified and analysis. However, the amplification of 400 amplicon is problematic because of the sample obtained is highly degraded. Hence, the distal 200 base pair hypervariable region 1 was therefore amplified. The sequencing of this region reveals high level of polymorphism within the population. In conclusion, the use of mitochondrial DNA and STR analysis made possible to type high level of degraded samples. However, the information content from the mitochondrial DNA may not as strong as STR analysis and nuclear DNA analysis, but it does provide the information from the distal identification team to differentiate the victims' remains. When matches could be made using mitochondrial DNA and confirmed by the STR analysis, we can say that the significance of matches are extremely high because of the population number was only 104. So I would like to say thank you for everyone for listening to my video, and I will also upload this to the YouTube and I will share the link at the comment section below. Thank you.